Welcome to this iGEM summer webinar. My name is Alexis Casas, and we're going to talk today about CRISPR systems. So I myself, I'm a PhD student at Imperial College, and I've been an iGEMer in 2018. I'm a member of the iGEM Measurement Committee, and I'll be giving this talk today with Geddes and Alicia, who are there. And now Geddes is going to start by talking about CRISPR systems.
Yep, thank you, Alexis. Hi, everyone. So I'm Geddes. I'm a PhD student at Imperial College. I'm at the Synthetic Biology Research Center, and I'm at the Biological Form and Function Lab, which is a new plant synthetic biology lab established at Imperial. So what is a CRISPR-Cas system, and what is the tool? So the tool itself has two components. The first one is the guide RNA, or the single guide RNA. And the second one is the CRISPR-associated endonuclease, and then it's referred to as the Cas protein. So the guide RNA is a short synthetic RNA, and it recognizes the target DNA region of interest. It directs the Cas nuclease there to the DNA for editing. It has two parts. Can you see my cursor?
Yes.
Yes. So the first part is 17 to 20 nucleotides long, and it's complementary to your target DNA. And the second part is the tracer RNA, and it serves as a binding scaffold for the Cas protein. The Cas protein itself is around 160 kilodaltons size, and it's non-specific endonuclease. That's why it needs the guide RNA to target the Cas protein to the DNA. And it makes double-stranded breaks at the DNA. There are many versions of Cas, and they're isolated from different bacteria, and we will talk about this later. So what is CRISPR? CRISPR stands for clustered regularly interspaced short palindromic repeats, and it's a part of bacteria's adaptive immune system against phage and conjugative plasmid infections. There are many steps of it, but it's quite straightforward. So it starts with foreign DNA entry to the bacterial cell, bacteria, and then the bacteria recognizes this foreign DNA. So it cuts repeat sequence and integrates it into the CRISPR loci. This CRISPR loci is present around like 40% of the known bacteria, so it's quite prevalent. So after it's integrated, it's transcribed. So there's a long primary transcript, and there is the tracer RNA, and that's constitutively expressed. These together combine and form the CRISPR, pre-CRISPR RNA, and then with the help of Cas proteins, the pre-CRISPR RNA is processed and matured into the CRISPR RNA. When Cas9 and CRISPR RNA interact, they become active, and if the target sequences are matched, then they cut the foreign DNA, and this is how the bacteria can remember, recognize, and clear infections. Yes.
So Alexis will explain the CRISPR history now. I realize the screen is on my side, so let me know when you want me to change slides and I will change it for you. Alexis?
Yep, give me a second. I'm just adapting. No, wrong panel. Yep.
Do you want me to stop sharing?
Maybe I could share.
Up to you.
Okay, no, we're gonna do it. Okay. Okay, I think we're good. Yep, should be good. So, I'm gonna talk a little bit about the history of CRISPR here, because you probably heard about CRISPR in the recent years, and I wanted to put back that into context to explain that it's not the work of just one single researchers and scientists wanting to target like genome editing, but it actually spans over 20 years of scientific research. And I want to give you a little bit of context before we start digging into CRISPR systems. So the discovery of CRISPR happened because it goes back to 1993, where a young scientist named Francisco Mojica from the University of Alicante in Spain started studying some microbes with extreme salt tolerance. And he found a very curious structure in a DNA fragment of a microbe genome. And it showed multiple copies of palindromic repeated sequence of 30 bases separated by spacers of roughly 36 bases. So what does palindromic mean, which is the P in CRISPR? Palindromic is when you have a word that you can read both ways. So for example, the word dad, D-A-D, you can read it from one side to another. The word mom, the word madam, M-A-D-A-M, you can read it the other way. So that's a palindrome. So it means that in the sequences of the DNA, we have the same thing. We can read the sequence from one end, from 5' to 3', or the other way around. So, lost my screen. Oops. Okay, and these did not resemble any family of repeats that are known in microbes. So he started looking at it and he investigated scientific literature. And he found a paper by a Japanese group, and that group had mentioned a repeat sequence found in Escherichia coli that had a similar structure, but not exactly the same similar sequence. And that was in 1987. And he kept on looking, and by year 2000, he found the CRISPR loci in about 20 different microbes, included Mycobacterium tuberculosis, Clostridium difficile, or the plague bacteria. And we go on to the next slide, please. Oops. Okay. So then he carried on working, and so he worked on trying to find these CRISPR sequences in genome for more than 10 years. And in about 2003, what Mojica did that he took each little spacer and he started to blast them with a blast tool. And he found out that one that matched, it was one E. coli spacer that matched the P1 phage sequence. And what he figured out is that that strain that was carrying that spacer was also resistant to the P1 phage infection. So he sort of established a relationship between those CRISPR spacers and the immunity system of the bacteria regarding the phage attack. And so figuring that out, he blasted about 4,500 spacers, and it turned out that 2/3 of these spacers matched known viruses or conjugative plasmids. So we're going to go on to the next slide, please. Okay. Now we're back. Now we're in 2002, and around that same time, there was someone else, Philip Horvath, who was working in the industry at Rhodia Food, and Horvath had been working on lactic acid bacteria. And at Rhodia, Horvath's work included developing DNA-based methods for precise identification of bacterial strains. And he was trying to overcome the frequent phage infections that affect industrial cultures. in dairy fermentation. So he was looking into this, and he wanted to understand how as Thermophilus strains were protecting themselves from phage attacks, and that he was doing R&D in this space because this has a lot of interest and economic importance for a company. You don't want, for example, like your yogurt fermentation being affected by a phage attack. And we're going to go on the next slide, please. Okay. So a few years later, the scientist John van der Oost from Wageningen University worked on actually programming the CRISPR. So what he did was inserting an E. coli CRISPR system into another E. coli strain that lacked its own endogenous system. And this allowed him to biochemically characterize a complex of 5 Cas proteins termed cascade. And he proved that the CRISPR RNA sequence were responsible for CRISPR-based resistance. So exactly what Horvath figured out, he managed to prove it. Then what he did is that he programmed CRISPR to target 4 essential genes in lambda-phage. And as predicted, the strains carrying the new CRISPR sequences showed resistance to the phage lambda. So he was actually the first scientist to directly program CRISPR-based immunity. Next slide, please. Wait, I'm lost in my window. Hello, Zoom. Where's my Zoom? Nope. Okay. Then in 2008, Mara Finney was convinced that CRISPR could not work by RNA interference because this mechanism would be too inefficient to overcome the explosive growth that happens when you have a phage infection. So it wouldn't be effective on RNA. And he thought that CRISPR must cut DNA like a restriction enzyme. And he actually proved that. So actually, it turns out that CRISPR can be used exactly as a programmable restriction enzyme, which is what we're going to see today on how to use this. So next slide, please. Then we go to a year later, we're about 2009 or 9, 10. We go to Canada, somewhere else, at the University of Laval in Quebec City. And Sylvain Moirot was studying CRISPR that cleaves DNA. And that cut, found out that that cut depends on the Cas9 nuclease. What it does is, so if the cut happens, you would figure out, like, you want to know why does it cut on the viral DNA, but not on the CRISPR sequence in the bacteria itself. And this is only possible because you have something called, where you have the blunt cut, you have something called the PAM sequence, which stands for the protospacer adjacent motif, which Alicia is going to talk later about. And it's a single blunt end cleavage. So you got the blunt end cleavage upstream of that PAM sequence. So this little sequence is in the viral DNA and not in the one in the genome. And so what Moineau did was that he found that viral DNA is cut precisely at the same position relative to the PAM sequence. And he also found out that when you have a number of distinct spacer, it equals a number of distinct cuts. And next slide. around the same time, Emmanuel Charpentier and Jörg Vogel were working on identifying microbial RNA. And to do that, they were doing massive parallel sequencing. And what they found out is that after recombinant RNA or transfer RNA, ribosomal RNA or transfer RNA, the third most abundant class of transcript is small RNA transcribed from sequence adjacent to the CRISPR locus. And there are 25 bases complementary to the CRISPR repeats. So they identify the tracer RNA. And next slide. So another researcher in Lithuania called Virginijus Siksnis was working on CRISPR system from the Thermopolis reconstituted in E. coli. And he found that transferring the entire CRISPR locus was sufficient to cause targeted interference against both plasmid and bacteriophage DNA. And what they did is they also proved that Cas9 protein is the only protein required for interference. So you only need the Cas9 protein to have the CRISPR system working. I'm going to go to the next slide, please. He then purified the Cas9 and CRISPR RNA complex. And he reprogrammed Cas9 with custom spacers in the CRISPR array to cut a target site in vitro. So he did some work in vitro to show that he could reprogram it. And he also showed that it was enough to just use 20 nucleotides for the CRISPR RNA and to get an efficient cleavage. And at the exactly same time, so we're in 2012, Emmanuel Charpentier partnered with Jennifer Doudna from Berkeley. And so they worked together and they used recombinant Cas9 and cRNA and tracer RNA that had been transcribed in vitro. And this Cas9 protein was expressed in E. coli from another organism. And like Siksnis, they showed that Cas9 could cut purified DNA in vitro and that it could be programmed with custom design cRNAs. And they showed that the two nuclease domains cut opposite strands that both cRNA and tracer RNA were required for Cas9 to function. So we now have these two RNAs that could function in vitro when fused into one single sgRNA, so single guide RNA. And now the concept of single guide RNA is now widely used in genome editing, and other scientists have made more modifications to make it work efficient in vivo. And next slide. So finally, in 2013, we get to what the wider public and everyone has heard about, which is genome editing in mammalian cell. And Feng Zhang was working on optimizing CRISPR systems. And in 2013, he published that paper where he showed that he targeted 6 insights in human and mouse genomes. And he showed that it was possible to mutate genes with high efficiency and accuracy. So you could do deletions via non-homologous end joining or inserting new sequences via homologous recombination. And you could also edit multiple genes simultaneously. Next slide. So here's a little timeline. So you can see that the whole discovery of all the CRISPR systems spans over 20 years. And I wanted to go through history because the discovery of the CRISPR is not only the work of a single scientist, but research from many scientists and in different countries that has spanned over 20 years. And the motivation wasn't initially to found a tool to edit genomes. It was a mix of scientific curiosity and industrial applications. and people just built on top of each other's work. And next slide. And with that, I wanted to thank you. And the next talk is going to be about experimental techniques on CRISPR systems, presented by Alicia.